Yeah, hope my voice is clear. Yeah, okay. My name is Qian Shi, and today I'm delighted to introduce our work Beaver, our method to accurately assemble transcript at a single cell resolution. In this talk, I will get you through why this problem matters biologically, and also the unique computational challenges we face, and how Beaver address them effectively. Many story in biology just begins with the central dogma. This fundamental process describes the flow of genetic information from the DNA, which hosts the genetic blueprint, to RNA, which acts as a messenger molecule, and finally to protein, which carries out the vast majority of functions in our cell. Specifically, a gene in the DNA is first transcribed into a precursor mRNA, which we call it the pre-mRNA. However, here is where the things get interesting. In complex organisms like human, this pre-mRNA undergoes a process we call it alternative splicing. Think of just like editing a film, different coding region, we call it exon, can be selectively kept or removed. As you can see here, by splicing the exons like the 1, 2, 3, 4, 5 here in different combinations, a single gene can be generated multiple distinct messenger RNA molecule, or to say the mRNA. Why this mechanism so important? Because this different mRNA, which we also call it transcripts or gene isoforms, can then be translated into proteins with different structures and consequently different functions. This process expands the coding potential of our genome from a limited number of genes. For today's talk, we will focus primarily on understanding this diversity at the mRNA level, figuring out exactly what transcripts are presented in the specific cell. There are two dominant RNA sequencing technologies, bulk RNA-seq and single-cell RNA-seq. As the name implies, bulk RNA-seq analyzes RNA that has been pulled from a mixture of many cells, like from a piece of tissue. This gives us an average picture of gene activities across the entire cell population. It generally cheaper and simpler, good for getting an overall view or compile the entire tissues. But it masks the difference between individual cells. If there are rare subtype or some subtle variants within the population, they get averaged out and might be missed entirely. On contrast, single-cell RNA-seq takes a huge leap forward by allowing us to profile the RNA content of individual cells one by one. As you can imagine, this gives us much higher resolution. We can uncover distinct cell types, track developmental pathways, and identify rare cell states that are invisible to the bulk methods. The trade-offs are higher cost, more complex network, and sparser data for each cell. There are different flavors of the single-cell RNA-seq technologies. The protocols of single-cell RNA-seq generally fall into two main categories. First, we have the droplet-based method, like those from the 10x genomics. These are high throughput and cost-effective, but typically only capture the sequence data from one end, either 5' or 3' end of the RNA molecule. This gives you what it essentially takes for each transcript, which is excellent for counting how many genes are active, but not for determining the full structure of the transcript isoforms. Second, we have plate-based protocols like the SMART6 series. While often low throughput, this method aims to generate reads that cover the entire length of the mRNA molecules. This full-length coverage is exactly what we need for detailed isoform level and analysis, and therefore this type of data Beaver primary targets. However, even with these powerful full-length protocols, single-cell RNA-seq data comes with its unique set of challenges. One major issue is dropout. While a transcript that is actually present in a cell might not be fully detected due to the technical limitations, leaving gaps in our data for that cell. In this example, you can see a gene with 5 exons at the top. Below, we have the transcript evidence from 3 different cells. In cell 3, for example, the evidence from junction between exon 3 and exon 4 is missing, this is a possible dropout event. Even when transcripts are detected, the coverage is often sparse, meaning that we get few reads for each transcript. And also, the PCR amplification bias can further distort the signal. This often leads to the fragmented transcript evidence within individual cells. The key insight of the Beaver leverage is that the information one cell drop out might still be presented in other cells in the dataset. Yeah, for example, like these missing junctions can be observed somewhere in the cell C1 and the C2. The central question for us becomes, can we borrow strands cross cells to fill in this kind of gaps without averaging away the unique biology of each cell? Okay, so let's look more about the dataset. Still for the type of the dataset, we mainly focus on the data generated by high-throughput short-read sequencing platform. These technologies produce million or even billion of reads, but each read is relatively short, typically 50 to 150 nucleotide long. A common technique is the pell end sequencing. We sequence both ends. This gives us two linked reads, we call it mates, separated by unsequenced gaps in the middle. We mark as the gray blocks here. For after sequencing, these reads are aligned back to a reference genome. This tells us the likely genomic origin of each read. Here you can see pell end reads mapped to a gene locus consisting of the 5 exons. These reads themselves may fall entirely within exons, or they might span across splicing junctions. The key challenge hinted here rests from the unsequenced region between the pell end reads and the fact that the reads themselves are short. This introduces the ambiguity. Let's go into the ambiguity problem, which is the center of the challenge of transcript reconstruction. First, with the pell end reads, the unsequenced gap between the mates is a problem. If the gap spans multiple exons or splice sites, we don't have direct evidence of the path that the transcript took between the two sequence ends. Secondly, the reads are short. They often cover only a small portion of the of an exon, like here, or might just span a single splice junction. Or if multiple transcripts show the same exon, a single short read could potentially match several different transcript isoforms. Let's look at this example. We have a gene also with the 5 exons. We also observe several parent reads. Now consider the top 2. Yeah, we marked as the pink here and on the top, and also the bottom 2 marked in green. If we assume the pink pell comes from the same transcript and is similar to the green pell, we might infer the pink reads supported a transcript using the exon 1, 2, 3, 4, 5, while the green reads support a transcript using exon 1, 3, 5. But what if the first read actually came from the same molecule with the last one? So I changed the color here. So as you can see that our interpretation could change completely. With only this fragmented reads, it's difficult to be certain about the full structure of origin transcripts that were presented in the survey sample. This changes from the nature of the current single-cell RNA-seq, and the short power and reads motivates the core computational task of our research, transcript assembly. The main goal here is to computationally reconstruct the full-length sequences of all the mRNA transcripts presented in a sample. The motivation is clear. The raw sequencing reads only give us the fragmented snapshots. It's like the little pieces of a puzzle. Transcript assembly aims to stitch these pieces back together correctly. I like to use the analogy of the assembly and jigsaw puzzle. The reads are individual pieces. Many of them might just look similar or ambiguous. The goal is to figure out precisely how they fit together to reveal the complete pictures, which in our case, the full-length biologically active transcripts. This includes both known annotated transcripts and also potentially novel ones. Why Is accurately assemble this transcript so important? First, it gives us much deeper understanding of gene expression complexity and the prevalence of alternative splicing. It reveals the true diversity of our molecules produced from our gene. Second, identifying the specific isoform presented in different conditions can lead to discovery of novel biomarkers for disease, uncover new regulatory RNAs, and even pinpoint the new therapeutic targets. Furthermore, this is especially crucial for this project. When combined with single-cell RNA-seq transcript assembly give us the insights into cell-specific transcriptional programs. As the figure illustrates here, by assembling the full-length transcripts within each cell, we can generate a high-accurate isoform-level expression profile for each single cell. So how do we computationally approach this puzzle? This brings us to the core of our method, Beaver. How does Beaver implement the idea of borrowing information across cells? There are many three stages of Beaver pipeline. The process begins with the stage 1, building a transcript fragment graph. This is the way we formulate this biological problem as a computational graph problem. Beaver starts with an initial transcript assembly performed for each cell. By default, we use the individual assembly from ALASH, but we also accept other assembly to provide inputs. In each input, transcript T has some associated coverage score. From this collection of fragments across all the cells, Beaver constructs a specialized graph structure. We call it transcript fragment graph. In this graph, each vertex here represents a transcript fragment observed in a particular cell, like the TC1 here, it means the fragment 1 in the cell 1. A directed edge connects two fragments, like here, saying from the TC1 in the cell 1 to the TC2 to the cell 2, if the suffix of the intron chain of the TC1 perfectly match with the prefix intron chain of the TC2. This suggests that these two fragments might be adjacent parts of longer transcripts. This graph, therefore, captures the potential connection between transcript fragments observed across the entire population of the cells. Now that we have this graph connecting all the small fragments, how do we identify the most likely full-length transcripts? This brings us to the stage 2. Well, we use dynamic programming to select a reliable candidate path through the graph. In the transcript fragment graph G, each path in the graph represented a potential full-length transcripts. To evaluate this kind of path, we first need to measure how well supported it is. For any given path P, we say transcript fragment T is compatible to the path P if it shares at least one splice junction with the path and does not conflict with it. We then define a junction weight for each junction J in the path. This weight is simply the sum of the coverage score of many compatible transcripts that support the specific junction. Our optimization goal is to find paths that maximize what we call the merging score. This score is the product of two terms. First, the AO is the bottleneck junction score. It means it is the minimum weight of any junction along the path. And also, the second term is the total number of junctions in the path. So why we have this kind of formulation? Because intuitively, this helps us to find paths that are not only long, but also consistently well supported across the entire length, avoiding some weak connections. This dynamic programming steps give us a set of high-quality candidate transcripts. But now we need to perform the crucial step of assigning them back to the correct cells. Stage 3 uses a machine learning approach to assign the final confidence score to each transcript cell pair. The ultimate goal of Beaver is to estimate the probability, let's call it PRPC, that a candidate transcript P derived from a path in the transcript fragment graph is both correctly assembled and expressed in a specific cell C. We approach this by using a two-stage machine learning strategy based on random forest models, with a set of carefully engineered features, 30 general features, and 21 cell-specific features for each candidate path. This general model predicts the overall likelihood that the path P is a correct transcript structure irrespective of adding specific cell. It acts like a coarse filter. Candidates scoring below a certain threshold like 0.2 are discarded. For the candidates that pass the first stage, this specific model provides a fine-grained score for each transcript cell PC. It predicts the probability that a transcript P is specifically expressed in the cell C. To evaluate our method, we design a set of experiments. We include two real datasets of human and mouse processed with the SmallSeq3 protocol. We also generate corresponding simulated datasets using RSEM to provide a controlled environment with the known grad truth. We use the data from chromosome 1 through 9 for training the random forest model. The remaining chromosomes are held out straighted as the test dataset. We compare Beaver's performance against a state-of-art tools. This includes some other meta-exemplars that also combine data from multiple samples like ALASH, Transmeta, and the SciClass, as well as some leading single-sample exemplars, Scallop2 and Straighthead2. Our grand truth for evaluation was defined as the follows. For real dataset, we used the trusted reference gene annotation to label our predicted transcripts to be the correct one or not. For simulated dataset, we have the perfect ground truth since we know precisely which transcripts were generated for each cell. As our primary evaluation metric, we used adjusted precision, which is calculated at the same level at the same level of sensitivity for each tool. This metric ensures a fair and direct comparison of how many false positives each cell produces when they are all identifying the same number of true transcripts. So let's look at the results. This plot compares the Beaver shown in the red points against the other exemplars. Each point here represents the performance for one cell in the dataset. The x-axis is the precision and the y-axis is the number of matching transcripts, indicating the recall. The vertical dashed line here indicates the average precision level achieved. Beaver consistently operates at a much higher precision at the same level of the sensitivity. For example, compared to Transmeta, Beaver achieves around 96.5% precision against Transmeta's 83%. This strong performance holds up on our simulated dataset as well. The story is quite similar, but there is an interesting observation. Transmeta, it is a meta-exemplar, ranked second on the real dataset, but here shows relatively poor performance on simulated datasets. Conversely, Scallop2, it is a single sample exemplar, did not stand out in precision on real datasets, but now it achieves high precision on simulated datasets. To investigate this kind of discrepancy, we analyzed two categories of matching transcripts. Cell-specific matches, which is the matching individual, which means those transcripts matching individual cell ground truths, and general matches, which means that those transcripts matching any transcripts in the collective ground truths across all the cells. For single sample exemplar like string type 2 and Scallop2 here, the general and the specific bars are nearly identical, which is expected because they analyze each cell independently. Now, look at the meta-exemplar, for example, ALASH. Make many total predictions, but it has a large drop-off to its general and cell-specific matches. This indicates it can correctly assemble some transcripts by pulling the data, but also produce many false positives. The key result here is the Beaver. Beaver starts with the same input as ALASH, but it dramatically reduces the number of false positives. That is the gap between the total the total prediction and the true matrix is much smaller. Beaver significantly improved not just the general accuracy, but also the cell-specific accuracy. It means that our learning framework is highly effective by filtering out incorrect assemblies and also correctly assigning transcript to the cell of origin. Let's summarize today's talk. First, we introduced Beaver, a novel cell-specific transcript assembler specifically designed for single-cell RNA-seq data. Its core innovation is its ability to intelligently leverage cross-cell evidence to overcome the data sparsity, while we use a machine learning framework to preserve cellular heterogeneity to avoid the averaging pitfalls of other meta-assembly methods. Beaver is open source and publicly available on GitHub at the following address. I would like to acknowledge those who made this work possible. I will first extend my deepest thanks to my co-author Qiming Zhang and our advisor Mingfu Shao for their guidance and collaboration. I also like to thank all the current and former members of Shao's research group for their support. Yeah, thank you for all your attention, and we'll be happy to answer any questions you may have.
Thank you for the presentation. We have time for one question. Okay, if there's no question, let's thank the speaker again, and also our speakers of the session.